Welcome to our panel on clinical gene and cell therapy trial design, where we'll be touching on regulatory strategy, innovation, and future directions of gene and cell therapy clinical trials. I'm joined here by a number of panelists. All of them have lived and breathed clinical trial design and execution, often basically being on call 24/7 to oversee these trials and make sure they're enrolled and the patients are taken care of well. This panel has developed, conducted clinical trials on several gene and cell therapy products, some of them which are now approved and on the market. So just to get started, we're going to play a little game of truth or dare. So the question that I have for the panelists, just to start off, is what's an example of, or what's the most exciting or interesting thing you've had to deal with or live through on a clinical trial? So let me start with John.
Yeah, sure. So thinking back early days, it was cerebral edema and severe ICANS that we had to tackle. So at Kite, as we were working through the Zuma studies, Zuma 1 being pivotal for Yescarta, Zuma 2 for Tecartus, early days we would get a number of severe adverse events, SUSAR type of events that we would get regulatory questions for. And it turned out that there was a deep desire by both the regulatory agencies and also the physicians conducting the studies to really understand mechanistically what was occurring. And so we got really good at developing biomarker packages and doing it quickly to meet 15-day turnaround times to demonstrate features of the product, features of the patient, the cancer itself that may have led to some of these adverse events. So, thinking back to those early days, we didn't really know what we were up against, but we learned very quickly and over time some scientifically what was occurring, leading to better intervention strategies. I think that the cerebral edema events were horrifying to us and very much a black box. And we had a couple of events occur while we were filing in Europe that needed to be addressed. And what we found from those studies were massive expansion of CAR-T, heavy production of noxious cytokines and chemokines, interferon gamma, high levels of IL-15, what have you, that led to multiple publications down the road, and I hope contributed to better management of patients for these types of adverse events.
So, Arati, I know that you'd worked side by side with John in some cases.
So John and I have lots of common stories where we've been up many nights stressing about patients, and so I don't want to kind of give out the whole IPD list on trials over here. It could get a lot of people into trouble. So I joke that I'm one of those people who could bike like 30 miles and not sweat. It's one of my, if you say what's the one thing about you, it's like one of my gifts, yeah. But I will say that the scariest and yet most exciting thing that happened for me was arguing for a label for patients that was wider than the trial that we had done. And when the idea first came up for Tecartus and mantle cell lymphoma, which had been studied only in post-BTKI relapse refractory patients, that's what we thought the label would be. But when we saw how great the results were and how much it could actually help patients, John was at many of those meetings, and I think I came out of every single meeting at the FDA and the EMA with I need to go and change my clothes because my pits are completely dripping because I'm so scared. And I think it was exciting because I think it's also a great lesson in the FDA listens to you if you provide rationale and you can show that this is going to help patients and you are going to take care of the safety piece of it. So that was truly exciting when the label came out to be a relapse refractory broad label, which basically meant it could help many more patients than the label we had studied.
Well, you're leaving out the funniest part is when you showed the label to the agency, the first question back was, this must be a mistake.
Yes, they did. So when I first put out the label, they said, is this a mistake? And I said, no, we have the data to show you. So that was the most exciting thing we did.
Yeah. Matt, you serve as clinical investigator, often principal investigator for many cell and gene therapy trials.
and I know you have hundreds of stories to share. Some of them I actually lived through with you, but I also know you've spent multiple sleepless nights trying to figure out how to take the best care of the patients.
Yeah, I think that's, one of the most exciting things is, giving a new investigational product to a patient who could benefit from it, and being the first patient, first dose, first patient on study, first time going after a new target. Can be a little scary at times. You don't know what to expect. You're afraid that things aren't going to work. You're afraid they're going to work too well. In the case of rapid expansion, cerebral edema, and ICANS. Just an example, I had one case where we gave the first in-human dose of a product, and I'm watching the lymphocyte count go from 0 to 1 to 2 to 4 to 6 to 8 to 12, all within a very short period. I'm going, oh dear Lord, what have I done to this person? And then, sending all these tests off, making sure there wasn't a transformation event, and then everything just contracting and the patient being in a complete response now beyond 2 years. So it's just the unanticipated outcomes, the unanticipated adverse events, and just trying to do the science in real time. So, we're working with people like John, when we were going through many of our IITs and trials, where we're trying to understand what we're seeing clinically in real time, get the information we need, and then treat the patient in the best way possible.
I do remember that, just for those who don't know, I think at the time you were worried that the patient had leukemia because
we gave them a
the
transformed cell or
lymphocytes were just blasting off. And I was on the sponsor IND side of it, and I was worried too, but it turned out that it was all CAR T expansion.
Correct.
And not leukemia.
Correct.
So yeah, there was a lot of sweating. Laura?
I think the thing I can think of is managing to finish enrollment on a phase 3 trial in the middle of a pandemic. So, that really was at a time when we were ramping up, and it was, something that you really had to figure out how to adjust to so many changing things. And we were very thankful for the many new guidances that FDA came out with during that time that helped us be able to continue going through our trial.
Yeah. I just realized we forgot to introduce our panelists, so if you guys want to squeeze in an introduction when you're doing your next answer, that would be great. So having lived through the development of not just conventional therapies, which might include small molecules and monoclonals, but then gene and cell therapies, but now more sophisticated next generation gene and cell therapies, the question I have for the panel is what have been your observations on how this has evolved, how you think about or how you've seen the clinical trials being conducted? So we'll start with John again.
Sure. So by way of introduction, I'm John Rossi. I'm Vice President, Head of Translational Medicine at Syncopation Life Sciences, which is a relatively young cell therapy company. Prior, I was at Kite for several years, and I led the biomarker clinical pharmacology teams on the translational side, supporting the approvals of CD19 CARs, as well as early IND development for new investigational products that Kite's going to be moving forward with. So to answer the question, prior years ago I was at Amgen, and we were working on small molecules, biologics. The studies tended to be fairly straightforward. Phase 1s were 3+3 designs into phase 2, and then larger phase 3 randomized studies. When I went to Kite, the thought was, well, it's going to be logistically impossible to run a randomized study, so we need to have single-arm study, relatively small patient populations that we were going to run as phase 2 to try to get full approvals. Back then, I think the key differences were the regulatory processes were being figured out. We grew up together with FDA and EMA to a certain extent. There was a lot of interaction that I was not accustomed to coming from larger pharma. There was a real interest from the regulatory agencies to I think get these products approved, and so we worked together across time during the protocol development, study execution, all the way through to filing to really iron out the processes. I think the key differences, and folks that haven't lived in the autologous cell therapy world don't fully appreciate, is how challenging it is to get a product to a patient and do it in a timely fashion. The infrastructure that's required from logistics, clinical, regulatory, is quite tremendous. So seeing commercial products now available to patients, to me, is still quite remarkable. But I think the key learnings are you have to separate, to a certain extent, cell therapy approaches from IO in general, and certainly from more traditional drug development. They're very different. The drugs themselves are living drugs. There's a lot of features that we need to understand. And I think, to maybe directly address Angela's question, the thing I learned and took away is make sure you invest in the science to support your clinical studies. It's not just, you know, the bread and butter PK, PD, do the cells expand, what kinds of cytokines do they make, but really more integrated scientific approaches that teach you about nature of treatment-related failures, nature of toxicities, and how you can use that information to think about generating the next better, or, you know, the next best product. So that's, you know, those would be my takeaways.
So, Arati, if you could introduce yourself, and I know you've already shared some of your experiences with the FDA.
Yeah, so I'm Arati Rao. I'm an oncologist, and I'm the Senior VP and Head of Clinical Development at PACT Pharma. Prior to PACT, I spent many years at Kite, where I developed Tecartus and took that to the FDA for mantle cell lymphoma. And then before Kite, I spent 18 years at Duke, where I trained, I'm a geriatric oncologist by training, and I spent many years doing the myeloid and acute myeloid and lymphoid programs over there. Okay, so for me, the kind of the big shift has been in cell therapy, having done this now for 6 years, is the interactions with the regulatory agencies, right? So I think it was very clear when we would go early on to the FDA, just the questions they asked us, the, you know, just the packages were just, you know, were a little more rudimentary compared to like when we started to go even a year and a half later, or when we went with the package. The questions were so much more savvy, and they know more, they expect more from you, and you know, that was a real clear feature. If you're going to say, this is what, you know, it's great if you can tell the FDA what you want to do, so long as you have the rationale and you put patient safety first, right? That's the biggest piece for them. And I think for me, you know, and I couldn't agree more with John, is that it's different from immunotherapy. You know, there's space for all different therapies being developed, and I think I'm reminded kind of when I was a fellow at Duke, where it was pretty clear when you did leukemia that you were a transplanter or a non-transplanter, and I think that's where drug development is going. You're going to be either a cell therapy developer or a non-cell therapy developer. Everything in cell therapy is different. ClinOps, the operations folks, don't have to worry about how you ship a pill, you know, but they have to worry about what LN2 do we ship the cells in, and how do we thaw the cells, and what that investigational brochure, you know, manual looks like. The regulatory interactions are different, manufacturing is different. Every single functional area is different in cell therapy, and I think that's the piece that I think, and it's moving so fast, and it evolves so fast, that I think the catch-up piece, I think, is the really, you gotta read the guidances, you gotta go to meetings like this to kind of, you know, catch up, make sure that you keep up with the latest and greatest.
Thank you. So we're going to go to Laura and then go back to Matt. But Laura, I know you've spent a lot of time thinking about oncolytic viral therapies and intratumoral delivery.
Yeah, so I'm a co-founder and former chief medical officer for Candel Therapeutics, which is developing an oncolytic viral immunotherapy for solid tumors. And in fact, the video that you saw before this session, that technology we in-licensed from Dr. Kioka at Brigham and Women's Hospital. And we, you know, I think that one of the things to point out is that one of the early approved products in the cell and gene therapy space was actually Imlygic, which is an oncolytic herpes virus, similar to that virus. And that's for metastatic melanoma. And I think the field has learned a lot about how to deliver viruses to tumors, and the advantages that has for being a really good way to not only kill tumor cells, but also to attract T cells into the tumor. So very good way to make tumors hot, inflamed, you know, tumors. And I think that's you know, been really interesting to see how much that has been working, and it also stimulating the immune system in a systemic way that can also lead to activity in distant tumors, the so-called abscopal effect. So it's even been seen in tumor types that are quite cold, like a glioblastoma. And I think one of the things that's also been important to learn from that is that when a tumor becomes inflamed, it will look bigger. And so you saw the example there where the patient, they thought the patient was progressing and turned out it was T-cell infiltration. So that's been really important for the field overall to build into the clinical trials that endpoints need to be thought about and tailored to the therapy, kind of like in the immuno-oncology space in general, that tumors may get bigger before they get smaller, even if they are actually responding.
So Matt, as the clinical investigator, you often comment on how science is moving a lot more quickly and regulatory sometimes just needs to try to keep up. So a quick intro and then
Hi, Matt Frigault. I'm the clinical director of the Cellular Immunotherapy program at Mass General. I kind of, looking back over a decade ago, I remember being in the lab when the first 3 CLL patients and the first BLL patient was treated with what eventually became Kymriah, and that was an immensely exhilarating time to really be getting the science coming to you in real time, coming off the LSR, looking at the cytokines, thinking about TOCI, and kind of getting that play-by-play. And that was a lot of fun and scary at times, but we did learn a lot from that. Fast forward to starting the program at Mass General, we had a handful of clinical trials, had treated maybe 30, 40 patients. Now we have upwards of 40 clinical trials, and we've treated hundreds of patients. And the amount of science and the differentiation of products, we're talking NKs, macrophages, iPSCs, T cells, iNKTs, gamma delta T cells. We're talking CRISPR, TALENs, zinc-fingered nucleases, lentiviruses, retroviruses. There's all these different technologies that we're starting to use. And, we set the rules, years ago and how we're going to operate. We just recently got some new guidance, at least from the CMC portion this last month. But overall, we set these rules, and then we need to constantly be adapting to the newer technologies that are coming. 15-year RCL testing in patients across multiple wide trials, looking at things, the large data registries. It's an immense amount of data. We started the program and we really modeled it initially like our phase 1 program for small molecules. And the head of that program was overseeing cell and gene therapy as it was growing. And that just turned into, we couldn't approach it the same way. It's just a different beast. There's more data, different types of trials, different degrees of reporting. Where else do you have to follow someone for 15 years even after they progress for, any adverse event that could be related to a product? This is really unique to cell and gene therapy. So my big take-homes are that when working with regulatory bodies, with building your own programs, and when dealing with the patients, the science is evolving much faster than I think we're accustomed to adapting, and that's something that we really need to be focusing on as this field continues to grow.
Can I add one thing, Angela? I think also for me, a lot of the guidances and things that we have learned so far have been from the heme malignancy space, which is great because heme malignancies kind of are the North Star of clinical development. But I think the big, having now spent 2 years in the solid tumor space, a very humbling 2 years, might I add, there's so much to do there. I think we've learned a lot, but I think there's just so much more that needs to be done for a group of 80% of patients who, we still don't have anything is helping a swath of patients across the board. So,
yeah, it's an excellent point. So I think a number of the panel's already shared a couple of sort of lessons learned that they took away from their experiences. Are there any other technical lessons, either scientific or operational, that you can think of that might be interesting to share with the audience? And we'll start with Arati.
Yeah, so for me as I think the clinical development / medical monitor person on a program, right? You represent the patient. You are you have the patient's best interest, and you represent that to every functional group when you work in cell therapy. And so I think, you have to work very closely with, every mat that you're working with at every site to make sure that you're holding that patient's hand right from the time that they sign the informed consent and when they get apheresed and their cells come to you, do when you dose them and follow them up and make sure that they continue to stay in remission, right? I always say that, I always heard manufacturing, there's a person behind this leukapheresis bag, so like when you say, oh, we can't leukapherese on a Tuesday, or we can't do this, no, there's like a gazillion things that need to happen to get this patient leukapheresed. They have 50 things that's going on, separate from their cancer, and then, we have to find a way to kind of fit that patient in and make sure that we can make this a great process, because they're doing us a favor and they're trusting us by coming onto this clinical trial. So I think for me, that was a big piece, that really kind of, working with every functional area. As the medical monitor, you also have to pay very close attention to what happens to the patient once you dose the cells, because if you don't like, that intense patient care, then cell therapy probably is not the right place to work in, because you really have to work. These patients get very sick, and you have to really kind of have your phone next to you and be there for these patients, because every patient is an N of 1. That's kind of what I've learned. There's been no pattern. If you ask me what a pattern is, I don't know. Every patient has been an N of 1. And it's people like John's teams who help me understand why patient A was different from patient B, right? So you're going to learn a ton from those first 10, 12 phase 1 patients. Make sure that you get the samples, make sure that you collect all of the biopsies that you need post-infusion, make sure that the AEs and the concomitant medications are all correlating, because otherwise the FDA is going to ask you questions on why you've labeled somebody a grade 1 CRS when in fact they think it's a grade 3 CRS. These are really small things, and I think the more you pay attention to that, the more you learn what needs to change in your protocol. That's what leads to a protocol amendment. Like, oh, we don't need to do XYZ, but maybe we should add XYZ in the ABC in the trial. And so that leads to a protocol amendment and makes it a better protocol. And a better protocol basically leads to better data entry and better data collection and a better study overall, right? So for me, that was a big piece, that, really working closely from start to finish, because there's a patient behind that bag, and you're only as good as your patients. So, yeah.
So Laura, while we were getting mic'd up to come onto this panel, you had mentioned to me some operational challenges or excitement that are typical for clinical trials, but maybe not apparent to those who have not worked on a clinical trial. So any technical lessons, either scientific or operational, from your perspective?
Well, I think one thing that's an exciting area that I think that our field in particular, could really benefit from exploring is the use of real-world data in clinical trials, and seeing how that can be used either to help, with understanding a disease better, to design better trials, or even as a supplement to a clinical trial to avoid having so many patients having to get placebo. And so this is something that I think the FDA has been coming out with guidances, participating in conferences on, and I think it's an area that for diseases that are either very lethal, where putting patients on placebo is really difficult and can be difficult for recruitment. It's something like glioblastoma, where the recurrent glioblastoma, the median survival is 6 to 8 to 9 months. That's pretty difficult to be looking at a placebo-controlled trial. So I think this is an area where also for orphan diseases, where really ultra-rare diseases where there are so few patients, being able to use the data that's out there for the natural history of the disease to support the regulatory and the clinical development process is really important. And I think what we've seen from FDA is that they're really focused on the quality of that data, and that being patient-level data that's collected either from health records or from control groups from other trials that have been completed, and focusing on diseases where it makes sense. So a disease like glioblastoma, where the natural course of that disease really hasn't changed, and the prognostic factors that are important are really well known, so that can really be done effectively. And so it'll be exciting to see how that may play out in the field as we move forward.
John, I think you had also mentioned to me in the past that you've been thinking about, or had to think about, real-world data as well, more and more. Can you comment on that?
Yeah, so I think I alluded to this earlier, and I'll focus on the KITE studies, but I think it's generally applicable to almost all of the CAR T products available now. commercially is that the studies themselves were small phase 1, phase 2, single-arm trials that led to relatively quick approvals. The challenge then for the translational scientist is that we're underpowered in those studies. We don't have the right comparators to really start to drill down on some of the biomarker findings or the hypotheses that were generated in those small studies. So the statistics never quite approach significance, or if you do get significance, it's, p equals 0.05, and that, people will roll their eyes depending upon who you talk to. So what you then need to do is look into the real world, where you're now starting to see thousands of patients treated with the 19 CAR T cells, so the 4 different products approved there, as well as the 2 multiple myeloma products, and open up access company-wide, and this becomes challenging from a commercial perspective, but also integrate the academic scientific community in allowing access to real-world data to really start to identify the predictive biomarker. And by predictive, I mean who should we treat, who should we not treat. And this was a lot of controversy at Kite, believe it or not, when we would come up with certain findings that would suggest, well, if you have high tumor burden, you're not going to do very well, maybe we shouldn't be touching those patients. But then you look at, there's overlapping patients that do incredibly well. They have durable CRs, very manageable toxicities. So it's not just tumor burden, for example. So the question then is, how can we predict the right patient for the right drug? And that requires access to real world data. And I think for anybody that's been working in drug development for a long enough period of time, whether you're giving aspirin or a very complicated small molecule or a cell therapy product, that you're going to see safety signals that emerge over time in small pockets. So recurrent cytopenias, for example, is something you see with the CD19 CARs, and I believe also is observed with multiple myeloma CARs. So the question is, how do we identify those patients? Because we can't get it with the 4 or 5 that we had in our 100-patient study. We need several hundred, and that's where access to real-world data and collaboration, both with academia and with pharma, I think is important to advance the field and to continue to improve upon the tremendous success that we've seen with the cell therapy products that are available to date. But it can't be done without broader patient data sets, more access to information, and that's where the real world, a CABMTR registry, things along those lines, I think are incredibly valuable, and we need to see more investment.
I'd just add one thing to that, is that I think the involvement of statisticians that have experience in this area is really important, and I think that there's a growing set of statisticians that are specializing in this, but there's nuances to how to do this, and I think that that's really will be helpful to see how that grows.
Good point. Matt, any other technical lessons, either scientific or operational, you care to share?
I think a lot's been said on this, but ultimately having a good relationship with the sponsor as an investigator. I think I'm texting many of the CMOs and medical directors, just making sure asking questions, going back and forth, asking for preclinical data, trying to understand what's happening with the patient. That's really helpful. But it's also really helpful to have a very lean and efficient machine when you're doing these types of trials. I say we're trying to build our program not to reinvent the wheel at Mass General, where we're trying to consolidate all these types of studies to a single group that has a diverse background of experience, solid tumor, heme malignancies, and even talking about with some of the autoimmune CARs that are coming out. How do we use the expertise that we've developed, the knowledge, do we spread that across the groups so that we're really efficient in how we do this with data collection, with AE reporting, with all the regulatory requirements that we have. So it's really about becoming efficient, having the good relationships, and we've worked with some of the sponsors too. We have a really good translational apparatus in our institution, and many places do. And so we've tried to leverage that, whether it be being able to get fresh cells on the 10x immediately after treating them, or running cytokines or doing additional panels that kind of go beyond what's already doing in the study. That I think has really taught us a lot about some of these more complicated mechanisms of toxicity.
So Matt, when you're enrolling into all of these clinical trials, what have you observed about the diversity of the patients being enrolled, and is that changing? Does that need to change more? What are your observations on that?
Yeah, no, that's a really good question. So when you think about how intensive a cellular gene therapy trial is, someone has to be able to come multiple times a week to see you. Someone has to have a ride. Someone has to have someone stay with you for an extended period of time. Just for a commercial product, it's the first 30 days. Some people are traveling from, really upstate Maine or coming from around the country for a clinical trial. I had patients on one trial in the middle of COVID flying from Chicago, California, Florida, Pennsylvania, definitely afraid of getting COVID, but also afraid of having their rare disease progressing. So, I think a lot of that has to do with, what type of patient are you accruing? Who has access to those types of resources? And what we commonly see is that we don't always get the appropriate representation of socioeconomic diversity, racial diversity, simply based on the limitations and what resources are available. I think companies are doing a better job at supporting patients, at providing housing stipends, at helping with flights. And that initially wasn't received well by the regulatory bodies. I think they were more afraid that that would be a form of, not bribery, but just secondary gain for the patients if they were giving a card for meals. But ultimately, that's how you get patients onto some of these studies when really they've exhausted every resource that they had leading up to this. They're multiply relapse refractory, haven't been working, have gone through their savings. There's a story of a patient from Australia. He actually sold his house to fly to the United States. That was a big thing that was in the news many years ago. So it's really just supporting these patients allows us to improve the diversity within these clinical trials, which one, helps you get a better idea of what would happen in the real world, because you're actually treating the patients who are going to hopefully receive the products. But two, you're also giving fair access.
And where does that support come from? Is it a combination of the sponsor, and if it's an industry sponsor, is it also the clinical site? So where does that kind of support come from?
All of the above. In many cases, I think the sponsors are helping where they can, right, through secondary foundations where they'll help manage the funds. We have hospital funds, we have donations, we have our nursing group runs this very large foundation, which is all donations, and it basically comes and I can call up and say, hey, someone had their echo delayed. Can I have a hotel room so I don't have to drive 4 hours back just to come back for an echo? And so we'll put them up, at the Wyndham or something down the street. But we really have to piecemeal this together, and it really became a problem with COVID because all of our Hope Lodges and American Cancer Society housing, even for Airbnbs who used to give discounted rates for patients, a lot of that shut down. So we really had to get creative with that, but I think having flexibility and looking into novel applications of some of these resources would really help patients get better access.
And just a question of curiosity for the panel. 10 years ago, when one would do a clinical trial budget projection, I've noticed that that number compared to what it costs now to do a similar clinical trial has almost doubled in cost. So the cost of per patient on the trial, which includes manufacturing, is that the same? Have you observed that increase or that spike in price, and what do you think that's being fueled by? So let me ask Laura.
I think it's definitely going up, and for many reasons, and now, with staffing shortages, that may even be adding to it. But I think that we also have the potential to influence that, to decrease that increase, I guess, by doing some novel designs and novel ways that we do our trials. So the FDA has come out with guidance on decentralized trials, and this is something that a lot of people are looking into. I think we learned from the pandemic that it's possible for patients to get some of their evaluations, if not even some of their treatments, closer to home by their healthcare providers that aren't necessarily having to come to the academic center for everything, and that can actually decrease cost and increase diversity by making the trials more available to a wider population of patients.
Want to add anything about the
Yeah.
cost?
I mean, the cost is going up. We see this across the board. Everything from get the right CROs to, your independent read, finding the right person to do it. It's more expensive, and so that's a piece. And I think in terms of the diversity piece that Matt spoke about, there's also the biological diversity in the TCR space. It's all HLA-A, O2, O1, and that's the Caucasian population. So to me, I mean, one of the reasons I love working at PACT is it is an agnostic platform. It's an HLA agnostic platform, and they cover all 6 HLAs, which gives, the Native American patient, the African American patient, the Asian patient to get on a clinical trial. That's a really big piece for me. And I think overall, it would be nice if there were therapies that were less toxic that we could bring to the population, because most physicians in communities can do apheresis, and if they can send the bag to the sponsor who can make cells and send it back to this physician, then they could infuse the cells locally and not have that patient come to an academic medical center if that product indeed was safe and they could manage the patient at their non-tertiary medical center, making cell therapy less elitist as it is today, right? Right now we serve a group of people who can make it to an academic medical center, and it really kind of needs to get to all patients, which is, I think, is going to be the challenge in the coming years.
I'll just add there real quick. There's not just so I always say, the ability to treat a patient away from a major academic center is a product of the toxicity of the product and the infrastructure that's available to treat that patient.
Yep.
And when you look at what it goes into treating these patients, of having a FACT accreditation, of having a larger research group, being able to do the 15-year follow-up, all of those regulatory components are actually somewhat restrictive on the smaller sites, because if they're only treating a handful of patients, there's not an economic incentive or even way to break even for them to have to get all their accreditations, train up their staffing, compete with CROs who are then recruiting the technicians and some of the data managers. And that's just a lesson for pharma out there. Giving all the money to the CROs to hire more staff is just taking away from the academic sites who are trying to recruit the patients. So remember that when you're working with your CROs. But you need to be able to make it feasible for sites to do a handful of patients that will increase access, so they're not having to go all to the larger academic centers.
I think on the point of cost, too, if you rewind back to 2014, 2015, when the first multi-center, company-sponsored trials were starting up, there wasn't an infrastructure CRO-wise to support specifically cell therapy. Through time, as you got commercial approvals, now you have all of the major CROs that cover your clinical trial or your biomarkers, what have you, all have specialized cell therapy divisions. I think with that comes a cost to have that specialization, which is another angle. And so you see it in how much does the lab kit cost, how much does the next-gen sequencing sample at your GLP lab cost, what have you, has all gone up. And if you're small, that also spills over then into your manufacturing. Who's making your vector GMP sourced or GMP grade? There's hot competition to get those slots. That's going to drive up the cost, right? Supply and demand. It's the same for who's making your cell therapy product. Once you have your GMP vector, because many times they're different facilities, now you have to make the cell therapy product. There are many quote-unquote vendors out there that specialize in this, but the good ones, again, everyone goes to them, and so again, it drives up the cost. So having this specialty boutique-like area of clinical development and clinical science, I think over time has, in a roundabout way, brought up the cost unnecessarily. As people say, we're cell therapy experts, we can support cell therapy, what have you. So that's critical, and I really agree with Matt's statement too on the small centers and the cost for them to treat 5 or 6 patients maybe on a clinical trial, or they have 10 commercial slots a year. It's not profitable for them to treat these patients.
You lose money.
Yeah, and that's a big problem for the patient, right? And something that I personally have a problem with, and I think we need to change. And this is where concepts of decentralized manufacturing may come into play. To Arati's point, can we develop products that have more favorable toxicity profiles so that patients don't have to stay for 30 days near the hospital and have a caregiver ready to drive them into the center should they have a late onset ICANS, what have you. These are things that I think are challenges to the field, but certainly the little guys suffer in the current model, and that's a real shame.
And I think one other thing is trying to minimize unnecessary tests that are included in our protocols. And I actually saw a presentation from FDA at the SITC meeting that was on this point, kind of tailored around what we've learned from COVID and how trials can be done more efficiently. So please take out the extra tests in your protocols that you're just copying and pasting that don't really need to be done for your particular trial. Don't do them. And that, I think, really does drive the cost up, and it makes it more difficult for the patients having to come in and get all these extra tests. And if there are things they're having done for their standard of care anyway, you know, trying to work through that in the protocol so that you're not adding extra things that they, at a time that they wouldn't have gotten their scan.
Let that PET scan from the week prior to consent
Yeah.
count for a baseline imaging assessment.
Yeah.
Yeah.
Allow that, yeah.
Yeah, we have a couple of questions that came in, and then we'll wrap up. One of the questions, this is for Matt. What is the hardest area to recruit patients if you were to think about it from a modality perspective or a disease perspective?
I think right now, solid tumors, mainly because one, you know, you need to get people excited. You have to have investigators and referring docs who believe in the therapy. Once they see their first patient go into a CR or have a durable PR, you know, that's an epiphany. All of a sudden, the referrals start coming. So letting people know that it works. On top of that, though, a lot of our technologies are A2 restricted, and we have to screen hundreds of patients in order to find an A2 specific and then an antigen positive patient. And so it's hard to, you know, the amount of work you put into some of the screening protocols, eventually it will pay out. You'll find the patients, you'll understand what works. You know, we're adapting and putting our HLA typing into our NGS panel. Mm-hmm. So rather than having a separate HLA testing, we'll screen everyone at diagnosis just like we look for KRAS mutations and other things like that. So I think we have to get better at that as institutions and as a system. But, you know, solid tumors, you know, I was just at IWCAR-T this last couple of days, and we had first day we had an entire day on CAR T cells and solid tumors and cellular therapies, and I think everyone actually was pretty excited. We were seeing a lot of activity. We were seeing patients who were actually having complete responses, and they're just anecdotes, but those are the same anecdotes that started CD19. So I'm hopeful in the next couple of years we're going to start seeing more and more of them.
Okay, great. Last question, and this relates back to our conversation about cost. Because it's so expensive to run these gene and cell therapy trials, do you think the future of cell therapies are bound to pharma? Does there need to be somebody with deep pockets? Does pharma have to be heavily invested and involved?
Who wants to take that one?
Well, as someone who's now in a startup, I'll say that, you know, the whole like getting, you know, patients excited to come on a solid tumor trial, I think it's getting investors excited too. It's like, you know, you do a phase 1, and phase 1s were traditionally, you know, known for safety, and now it's like, oh, you don't have a PR, you don't have a CR, we won't give you money. Well, you know, like, how do you go on, right? So that, I think that's been kind of the eye-opening piece for me, is that, and then you show a CR and they're like, oh, well, we want to see duration of remission. And it's like, wait, this is not a phase 2. This is a, you know, phase 1 study. I'm showing you that this data is safe, and, you know, we're seeing these cells traffic into the tumor, which we've never shown in, you know, solid tumor space. I think that's the really hard part. So I think I do think that the cost really is very high in the TCR space, because you just need to make a whopping dose of cells in the solid tumor space, not the, you know, the small doses that we do in heme malignancies. And so I think the COGS are just so high. You do need someone to come and support you, whether it's a really big investor versus, you know, pharma coming in and sweeping in and saying, we like your platform, we like what you're doing, you know, let us help you help patients.
But I think technology is going to help with that, right? Once we move away from lentiviral vectors, we move into more advanced gene editing techniques that may not be as expensive. You know, we have to find a way to make this work. These responses are too compelling to just ignore them. And so I don't think cell and gene therapy is going anywhere anytime soon. Or at least I hope not, because I'm banking my career on it.
I don't think
but
so.
Ultimately, I think technology will get us through there.
That's a perfect way to close. I wanted to thank the panel for sharing your insights, and thank you to the audience for participating.